Hello, and welcome to our discussion on more complex Mendelian genetics. Now, it's going to be a little bit more in depth than the simple Mendelian genetics that we've been dealing with in the past, but I promise that it is going to be interesting because we are going to meet a dog named Sessa. Now, Sessa is a German Shepherd. German Shepherds were originally bred in Germany, and they were bred to be herding dogs. Now, Sessa's a long way from Germany and her home in California, and she does not do any herding, but she loves to lay upside down, celebrate birthdays, and wear fun hats. Now, Sessa was adopted when she was 1.5 years old by her human parents. Sessa's human parents adopted her from the shelter, and they never knew her dog parents. So, Sessa does not know what her dog parents looked like. Now, the question that we need to answer today is, could Sessa's human parents, using their knowledge of Mendelian genetics, determine what Sessa's dog parents could have looked like? Is that something that we can do? Now, to answer that question, first we're going to have to review Mendelian genetics. Now, Mendelian genetics were brought about when Gregor Mendel observed some differences in pea plants. Now, when he did that, he started to breed the pea plants and further his observations, and his hypotheses and eventual theories are as follows. Now, he stated that Mendelian traits are traits that are passed down from a parent to an offspring. So we're talking about physical characteristics here. Now, how does this happen? How do peas, like we see in our little GIF here, pass down their traits to their offspring peas? Or in this case, how would Sessa's dog parents pass down their colors? Now, they do this through sexual reproduction. Sessa's dog dad, or this dad pea, would pass down one allele from their color to their offspring, so to Sessa. And Sessa's dog mom, or in this case, this yellow mom pea, would pass down one allele of her color to her offspring. When these alleles combined, they form genes. Now, our genes are found encoded in our DNA, and they're like little instruction manuals that tell us all kinds of things about our body. And in this case, they will tell Sessa what color her fur will be. Now, if we want to try to figure out what color Sessa's parents were, let's first talk about Sessa. Now, she is German Shepherd, as we talked about, but she is specifically a bicolored black Shepherd. We know this because she has an all-black body and face, and she has tan legs. Those are key characteristics of our bicolored black Shepherds. Now, do all Shepherds look like this? No. We have a variety of different Shepherd colors, and our three main ones that we're going to talk about are our sable Shepherds. They are characterized by being a variety of different colors, so liver or blue, and among other things, but all of their fur has a black tip, and that creates kind of this all-over dark look. Then we have our black and tan Shepherds. These are the Shepherds that you probably are most familiar with seeing, and their body is covered with characteristic patches of black and tan. These contradict our sable Shepherds because you can tell the tan on our black and tan Shepherd is just tan and there's no black on the tip of that fur. And lastly, we have, like Sessa, our bicolored black Shepherds. They have mostly all black bodies and faces with tan legs. So, now that we know that Sessa is a bicolored black Shepherd, let's talk about our genotypes. Now, those colors that we just mentioned, those are all of our phenotypes. That's what we can see. We see in our German Shepherd sable right here that we have that black tipped fur. That's our phenotype. Now, what genotype characterizes that? We have our genotype, capital AW, capital AW. We have a potential genotype of capital AW, capital AS, and we have a potential genotype of capital AW, lowercase at. Now, the first thing you're probably noticing right here is that we have 4 letters instead of 2, and we are used to dealing with 2 letters in our simple Mendelian genetics. Now don't worry about this, we're going to talk about this and we're going to talk about our dominant relationships. In the meantime, the most important thing to note here is that in our sable shepherds, we see an AW in every single one of our genotypes. So we know that our AW is going to be strongly correlated with our sable shepherd. Okay? Then we will move on to our black and tan shepherds. Black and tan, again, that's going to be our phenotype. Our genotype that we're going to have are capital AS, or we have capital AS, lowercase at. Now, we know that when we have our dominant traits in simple Mendelian genetics, we are talking about our capital letters there. And when we have our recessive traits in our Mendelian genetics, we are talking about our lowercase letters there. In this case, we're talking about that first A. That first A is capital or that first A is lowercase, okay? I want you guys to make sure that you are taking note of this as we're talking through it, and we're going to explain the more complex dominants in a little bit. But make sure that you're noting these things so as we work through, we can use context clues to really figure out where we're going with this. Now, what similarities do we notice in our black and tan German Shepherd genotypes? That's going to be our AS. So we have AS and our ASAT, both of which have the AS, so we know that our black and tan is going to be coded for by our AS allele. And last, we have our bicolor black German Shepherd. Our bicolor black German Shepherd is only going to have the potential genotype of lowercase at. Okay? So, now, like I said, we were going to talk about the more complex 2 letters. All this means is that our alleles have 2 letters encoding for them. So, when Mendel first hypothesized Mendelian genetics, he thought that simple Mendelian genetics was really what existed, and we had 1 letter for each allele, and these 2 alleles would combine to form a gene. Now we still have 2 alleles that are combining to form a gene, but our alleles are being encoded for by multiple letters. Okay, so this is where we're kind of stepping in to our more complex Mendelian genetics. So, German Shepherd fur obviously has multiple alleles. We can see that because there are 4 letters instead of 2. And we got to remember all the time that science is dynamic. Science is constantly evolving and changing, and we're learning new things, especially as new technology comes into our hands. So, super important to remember that even though Mendel hypothesized simple Mendelian genetics, we are changing, we are being dynamic, and we are working our way into these different possible dominant relationships that are being allowed for by multiple alleles. Multiple alleles, as you see in our GIF with the P's, and multiple alleles like we're going to talk about today with our German Shepherd. Now, when we have multiple alleles, we're obviously going to have a complex dominant relationship. And this is where we're really going to get into it. So, we know that we have, first of all, our AW here. This AW right here, we know encodes for our sable shepherds. So we remember our sable shepherds right there. Then we move on to our AS. We know that our AS encodes for our black and tan shepherds. So let's take note of that. And lastly, we are going on to our AT, and we know that our AT is our bicolor black shepherds. Now, remember, as we're working through this, we're keeping that main question in mind, and that main question is, Sessa is a bicolor black shepherd, and we are looking for combinations of parents that would create a bicolored black shepherd. Now, to do that, we need to establish these dominant relationships. So, the first thing to note is our AW, our sable shepherds. That is going to be completely dominant over everything else. So AW is going to be dominant over AS, and AW is going to be dominant over AT. That's our complete dominance. We're really familiar with that from our simple Mendelian genetics. Okay? Then we're going to move on to our lowercase at. That is going to be completely recessive. So completely recessive, we're really familiar with that from our simple Mendelian genetics as well. That means that if paired on a gene with an AW or an AS, it's going to be recessive to that allele. Now our recessive alleles and our recessive traits are only going to express when they're combined with another recessive, right? So anything with these dominant ones is AW or this AS, and we're not going to see the AT. Remembering, our goal is to find those bicolored black shepherds where we see the AT, so that's going to be important. But then we have this AS in the middle. So how do we figure out the dominance and recessive relationships of this AS in the middle? Let's break it down, okay? So if we remove our AW from the situation entirely, we know that our AS is going to be dominant over our AT. Okay, so we know that this right here is the dominant relationship. Now, if we go the opposite direction and we remove the AT from our situation altogether, we know that our AW is going to be dominant over the AS, making our AS recessive. Okay, so we've had one situation where the AS can be dominant and one situation where the AS can be recessive. And that is what we're going to call incomplete dominance. Okay, so we're just doing a little basic introduction to incomplete dominance today. But just know that Mendel did not theorize incomplete dominance in the beginning when he was working with his pea plants. And it's come about because we're constantly studying science and we're constantly figuring out what's new, what's changing, what we can correct. Okay? So our sables are going to be the most dominant as a quick review. Our bicolored blacks are going to be the most recessive, and our black and tan is going to be a little bit in the middle, so we're going to have to pay a lot of close attention to each allele in each situation to see what our offspring will be. Okay? So we know that Sessa is a bicolored black shepherd. We talked about this. We're comparing her to our other bicolored black shepherd there, and they both have black faces, black bodies, and tan legs. So we know for sure that Sessa's genotype is that lowercase at, because we know that the bicolored black shepherd is only homozygous recessive. We know that those are her genes right there. So that's what we're looking for. Keeping this bigger question in mind, which parents can produce an offspring with Sessa's genotype, lowercase at? So how are we going to do that? Well, let's look at our genotypes of our potential parents. We have our sables with those dominant AWs. We have our black and tans with our incompletely dominant ASs. And we have our bicolored blacks with our completely recessive ATs. Okay? And I know this is overwhelming, but here are our 22 potential parental combinations for Sessa. This is a lot that we got going on here, but we're going to work through with a few Punnett squares, and we're going to figure out some hints that are going to help us to get to the bottom of this question here. So these are our potential parental combinations. We don't have to go over them all now because we're going to do one at a time. So we're going to work through our first hypothesis. So if Sessa is a bicolored black shepherd, figure might as well just start with a bicolored black shepherd combination. We know that their only potential genotype is going to be a lowercase at. So our parent 1 and our parent 2 are going to both be homozygous recessive bicolored black shepherds, and we're going to work through that Punnett square. So for our Punnett square, we are going to make sure that we are putting the genotype of our first parent on top. Now you see how we are splitting up the alleles there. When we have 4 alleles, or when we have 4 letters that are making up our alleles, we still have those 2 alleles. So it's going to be AT as 1, and our other AT as the other, okay? And then we're going to move on in pink, and we're going to put our second parent there on our side. Now remember that when we're working through Punnett squares, it's really important to be pulling one of your alleles from that top, that's going to be your first parent, and one allele from the side, that's going to be your second parent. Now no parent is just going to give two alleles and not have any other alleles from the other parent to the offspring. So it's really important that we make sure we're taking one allele from each parent to create our offspring. Okay? So as we're finishing up this Punnett square, we can see that our homozygous, recessive, bicolor black parents that are both genotyped at lowercase at, are making 100% offspring, lowercase at. That means that 100% of these offspring are going to be bicolor black. Is that what we're looking for? Yes. So we're looking for a parental combination that will create bicolor black offspring, and right here we have one. So we know for sure that these two colors of dogs could have been Sessa's parents. Now, is this the only combination that we could have? I don't know. So let's move on and see. So our best way to check if that's the only combination that we could have is to move on to another hypothesis. So we're going to go with our hypothesis number 2. Last time we tried homozygous, recessive, bicolor black shepherds. So today we will try Sessa's dog parents both being homozygous, dominant sable shepherds. So we have homozygous. Remember that homozygous means we have two of the same alleles. So AW, that's going to be really important. And we have our dominant sable shepherds. We know that that AW is going to be completely dominant. So we are going to note right here that we have our genotypes. Something to think about. We're going to work through this Punnett square. Something to think about here is, do we have a lowercase at to pass on to a bicolor black shepherd? So as we're working through that Punnett square, let's think about that. If neither of our parents have a lowercase at to pass on to our offspring shepherd, are we going to end up with an offspring shepherd that is lowercase at? I don't think so. We're going to work through our Punnett square because it's important that we are checking our work and that we are making sure that when we hypothesize a hint, that we really do the work to see if we're on the correct track. So we started our Punnett square by putting our parent 1 up top with our uppercase AW, and then our parent 2 in pink on the side with our lowercase AW, and we're working through the Punnett square like we always do, pulling one allele from each parent, and it looks like we are going to end with 100% of our offspring with the genotype of AW. Now we remember that phenotype is going to be our sable shepherds. So if we are looking for a bicolor black shepherd, is this a potential combination for Sessa's parentage? No. So we know for sure that Sessa's parents are not both homozygous, dominant sable shepherds. And so we're eliminating some stuff out. So let's think about what we just learned there and kind of how we can apply that to our Punnett squares going forward. So we talked about if we have two homozygous parents, homozygous dominant parents, in this case of the sables, are we going to end up with a homozygous recessive offspring? And that's going to be a no. If our parentage generation does not have any recessive alleles to pass down to the offspring generation, our offspring are going to be all homozygous dominant. So how does this help us when we're going through our potential parental combinations? Okay, Well, let's think about this. We started with our bicolor black combination right here in the corner, and that left us with a positive result. We do have potential for recessive parentage there because we know those are homozygous recessive. But then we moved on right here to our sable homozygous dominant option. Did that give us any parentage? No, definitely not, because we did not have the ability to pass on that recessive trait. Now, what other one can we eliminate here if we're eliminating this homozygous sable option? We can also go through and we can eliminate this black and tan homozygous option. Now, we can do that because we know that those are going to be homozygous dominant, right? And we're looking for homozygous recessive. If those parents don't have a recessive trait to pass on, there's no way that we can end up with a recessive offspring. Now, this is going to take our potential parental combinations down from 22 to 19, okay? So, let's work through hypothesis number 3. As we're keeping our hint in mind, we're not going to do any more homozygous dominant combinations because we know for sure that we are not getting a homozygous recessive offspring from that option, okay? So for number 3, let's try cis dog parents being one heterozygous dominant sable shepherd, showing an AW-AS genotype, and one heterozygous black and tan, showing an AS-AT genotype. Now, we know for sure that we're getting closer here because we have this AT right here in our parental number 2. So we know that we have a recessive trait to pass on. So is this going to work? Let's think about this while we do our Punnett square. Set up the Punnett square just as we usually do. We're going to take that first parent with the AW-AS, that is our homozygous, our heterozygous dominant option for our sable shepherds, and then we are going with our AS-AT in the pink, so that is going to be our heterozygous recessive option for our black and tan shepherds. As usual, we are going to pull one allele from the top and one allele from the side as we work through this Punnett square. Now, something to note real quick, it is a great idea to do your Punnett square with your parents in different colors. As we go through and we add more alleles and more genes to this complexity that is Mendelian genetics, it's going to be a whole lot easier if we can color code. So, now that we've finished our Punnett square, we are going to separate out the offspring. So here in orange, we are circling our sable offspring. So we see that dominant AW. That dominant AW is always going to give us that sable. Then we have here our green, and those are going to be our black and tan offspring with that partially dominant AS, right? So something to look at there. We do have some recessive alleles. However, look at what our recessive alleles are paired with. Our recessive ATs are paired with a dominant AW and our incomplete dominant AS. That means that we will not see those recessive traits, those lowercase ATs. We will not see a bicolor black offspring. Okay? Here we are ending with half of our offspring being sable and half of our offspring being black and tan. We know for sure that we are getting closer because we are ending with some recessive alleles, but we know that we are not quite there yet. Okay, so what are we going to do in science when we're not quite there yet? We're going to reevaluate our hypothesis. We're going to edit it a little bit and we're going to work through. So for hypothesis number 4, let's try cis dog parents being one heterozygous recessive sable shepherd. That's going to be our genotype of capital AW, lowercase at, and one heterozygous black and tan shepherd. It's going to be capital AS, lowercase at. Okay, so as we're working through this, setting up the Punnett square just as usual, let's think about what we're seeing here. So in black, we have that first parent. We have that AW, that dominant allele right there, but then we have an AT, that's our recessive allele, that's what we're looking for. Then as we set our second parent up in pink, we have our incomplete dominant AS. In this situation, it's acting as dominant, obviously, because it's matched with that recessive AT. So this is the first situation, other than our homozygous black bicolor shepherds, where we have 2 AT, two of those recessive traits that we're looking at. And as we complete our Punnett square, we can see that because both parents had a recessive trait to pass on, we are ending with one fully homozygous recessive option. Okay, so let's go through and let's categorize our offspring. We're going to have 2 sable shepherds. We're going to have 1 black and tan shepherd. And we are ending with one that is the one we are looking for. We are ending with 1 homozygous recessive bicolor black offspring. So the first thing that we can note is that we have 50% of our sable shepherds, 25% of our black and tan shepherds, and 25% are going to be bicolor black shepherds. First things first, is this a potential parentage for Sessa? Yes, absolutely. Even though we started with a sable and a black and tan, 25% of those offspring are going to be bicolor black shepherds. And that's great news for us because that is exactly what we are looking for. Now, just a quick thing to point out, you see in our black and tan shepherd right here, we have our AS as our second allele, and then we have our AT here as our first allele. Now I know that we're used to seeing the capital letter, that capital AS in the front, so we're used to seeing that flipped. However, it's really important to know that even if it's flipped, that does not change our dominance order. Our AS is still going to be dominant over that lowercase at. So even though they're flipped, we're still seeing a black and tan shepherd there, and that's really important to note. Okay? So potential parents for Sessa, we are getting way closer to answering our question here. And let's think about what we just saw. So we just saw 2 parents that both had recessive alleles to pass on. So 2 parents that are heterozygous, represented here by our cute little peas, and they were able to create a variety of offspring, at least one of which is going to be homozygous recessive. Now that's really, really important as we're moving forward, because we know now that to get that homozygous recessive offspring that we're referring to, we definitely need both parents to have a recessive allele to pass on. So in the context of our question, what's that going to mean? So we have our 22 parental combinations, right? We know that our homozygous recessive bicolor black combination, that's going to be good, that's going to work. We have already eliminated our homozygous dominant combinations because we know for sure those aren't going to work. Okay, then we came through and we eliminated all of the ones that did not have one recessive allele to pass on. Eliminated them all. Now, with the hint that we just talked about, we know that we are going to need to eliminate every single option that doesn't have 2 recessive alleles to pass on, right? 1 from each parent. That takes us down to 14 parental combinations. Okay, so we have now eliminated every single option where we do not have 1 recessive allele from each parent to pass on. If you look at what we have left right here, you see we have an AT and an AT. That's 1 recessive allele for each of our parents to pass on. right? This is that example that we just did right there. That is our one recessive allele for each parent to pass on, and we now know from our trial and error, from our hypotheses, that we need those double recessive alleles. So look right here, double recessive, double recessive, double recessive. Each parent has that potential to create an offspring, okay? That's really important for us to note right here. So we're going down from 14, which we originally had when we eliminated that first round. So we're going down to 5 potential parental combinations that we are working through. So we feel pretty confident in where we are, but this is science, so we're working through, we're doing another hypothesis as we kind of adjusted it for our hint, and we're going to work through another option to really know that our hint is going to be correct in all situations, okay? So our hypothesis number 5, the last hypothesis for today, is that Sessa's dog parents are one heterozygous recessive sable shepherd. So heterozygous recessive is going to be our capital AW, lowercase at, and one homozygous bicolored black shepherd. It's going to be our lowercase at. So let's work through that Punnett square. As usual, we're setting it up. Parent number 1, that's going to be our heterozygous recessive sable parent with that capital AW and then the second allele of the lowercase at. Then we're going to set up our second parent in pink on the side, and that is going to be our homozygous recessive, because that's the only way they come, bicolored black shepherd, that lowercase at. We're going to go through, we're going to take alleles from each parent, and we're going to work through our Punnett square here to see if that hint that we came up with is going to be correct. Now, as we are completing our Punnett square, we are seeing that we are for sure getting some homozygous recessive offspring. So when we categorize them, we see that we have 2 sable shepherds because we got that dominant AW that's always going to express over our recessive AT. And we are ending with 2 homozygous recessive bicolored black shepherds. That is 50% of our offspring sable, 50% bicolored black. So is this a potential parental combination for Sessa? And the answer to that is absolutely. So here was where we left off. We had our 5 parental combinations. We tried this one up here in the corner. That's the one that we talked about first. And then we confirmed our hint by trying this one right below us, that sable bicolored black. That's a Punnett square we just did. And we are left with these 3 potential parental combinations left. Now we formed our hypotheses. We formed our hint. We really thought through the science. And we really thought about why our hint would be correct. Why would it be important for both parents to pass on at least one recessive allele? And when we thought about that and we talked about that, we felt confident in it. We tested it with one more hypothesis, and so now we're sure. We feel good about the fact that all of these potential options, with each parent having at least one recessive allele to pass on, is going to be a potential parental combination for Sessa. We know that because we're confident in our scientific method and our process. And we see that all of these circled and highlighted options here, the highlighted ones being the ones we tried, and the circled options being the ones that we are applying our hint to, we know that those are going to create our homozygous recessive bicolored black shepherds. Those are our 3 potential combinations left. And here is our conclusion. So this is everything that we've worked through. We have our hypotheses 1, 4, and 5. Those are the Punnett squares that we did. Those are the ones that we worked through. They left us with either 100% bicolored black offspring, 25% bicolor black offspring, or 50% bicolor black offspring. So those are all potential combinations for Sessa's parentage. And then we have these 3 additional. Those are the ones where we took this hint here that we figured out when we worked through 1, 4, and 5 and their Punnett squares. And we worked through 2 and 3 that didn't work out but got us closer to our correct answer, and we applied them to these additional hypotheses right here. And we know that just from that information that we are going to end up with 25% offspring, 25% offspring, and 50% offspring that are all going to be bicolored black shepherds. Now, we can always do the Punnett squares to check those, but we know for sure, based on our science and based on our hypotheses and our hints, that that's what it would work out to. So here we have highlighted our 6 potential parental combinations. We know that these are the combinations that could create a shepherd like Sessa. So back to our original question, which was, can Sessa's human parents figure out what color her dog parents might have been using the principles of Mendelian genetics? Now let's think about that for a second. Our answer is going to be kind of. So can Sessa's dog parents definitely know some potential combinations of parents? Absolutely. We know that her parents could have been 2 homozygous recessive bicolor black shepherds. We know that her parents could have been 2 heterozygous recessive sable and black and tan shepherds. We know that there are some options, but we don't know for sure which color her parents were, but we did eliminate a lot of potential. So our answer to this is kind of. Can they kind of figure it out? Absolutely. Did we narrow it down? Yeah. And that it was a great use of practical Mendelian genetics. So Sessa says thank you for helping her narrow down her biological parents a little bit, so she knows a little bit more of where her cute looks come from. Now Sessa is very happy with her adoptive parents, but it's always important to know a little bit more about your genetics and your biology. It gives you a little power and a little strength. So these are our next generation science standards that we covered in this video, all about heredity and the inheritance and variation of traits, all in the context of Mendelian genetics. Something that we also really touched on again as we work through the year. We're always going to come back to our scientific method, so that observation, questioning, hypotheses, experimenting, concluding, fixing, theorizing, all of those things that are really, really core to our science, that's how we're going to figure everything out. So we really hit on our scientific method in this context as well. We worked through some practical examples, we worked through some models, we worked through some writing and some visuals and some videos. And this really is getting us closer to just a solidified, complete understanding of our next generation science standards and how we can apply those to our science world, to the other topics that we're studying, and just really solidify all of that knowledge. So here are our notes and our sources, and thank you guys so much for working through this with me and for helping Sessa out.